Okay, good morning, everyone. I'm really thrilled, and actually I find it quite breathtaking that we have so many people here in the audience. It's getting me, to be honest, a bit emotional. Finally, after 2 years, we can manage to have this Congress with all of you here, so many people from so many different countries. So I'm really amazed that we just pulled it off. So thank you for joining. Frank? Thank you, Gaby. Thank you. So I want to thank Professor Frank Kooy for really organizing this here at the University of Antwerp. And a special thanks to Claudio, who will give later on a presentation as well. He is one of the PhD students within the team of Frank and actually did all the practicalities for this event. So thank you, Claudio. So just to give a bit of an introduction of myself, so I'm Kelly Verbruggen, I'm the mom of Cedric, and actually he was, so to say, patient zero in the whole discovery of the ADMP syndrome, which is better pronounceable than saying Helsmoortel van der A syndrome. Every time we need to repeat it over and over again. What is the syndrome? So, okay, it's just ADMP syndrome. It's much easier. So my husband and myself, we started our journey at the birth of Cedric in January 2009. And from day one, I really do believe that as a mom, you have a kind of a sixth sense. You feel that something is not okay, something is wrong, but you can't put your finger on it. And it took me actually 8 months before a pediatric doctor really just came to me by himself, like saying, okay, I see some dysfunctions in your child, so you really need to see a genetic doctor. So, and that brought me to Nathalie van der A, who actually lives 2 streets away from where I live. So what a coincidence, no? And then the journey started, and it brings us to here. So actually we are now with a whole bunch of people on this journey, and I hope that this day will bring you some more insight on the syndrome that our kids all share. So therefore I would like to give the floor to Professor Frank Kooy, who will give an introduction further. Normally it would be Nathalie van der A herself who would be present, but unfortunately she's ill, so she could not join. So Frank, the floor is yours. Thank you.
Well, thanks so much for organizing this all. Thank you all for being here and for coming. Just 2 switches on and off. This is the right one. Thank you all for coming here. I will first give you a little bit of background on the meeting. Why? Kelly is actually organizing the patient day, but why? What's my involvement in here? I would like to do that just by showing us the sponsors. And the sponsors, which are gratefully acknowledged, and these are also the reason that we can keep the price, which we think is very affordable. Sponsoring has been very, very generous. And what you see, and before this, we had a 2-day scientific meeting. And how were these people selected? Well, about half of them, they were part of 2 European networks, an ERANET Neuron and an eRare network. What is that? That is EU-sponsored projects that are given to small consortia, so not the European, not the very big ones, but the smaller ones, and that allows us to work on topics that are sort of hot at the moment. The moment we supplied, so the ADMP gene was hot, so this is one of the reasons why we're here. We've also been sponsored by the Scientific Foundation, a local charity, the European Society for Neurochemistry, the ADMP Kids Foundation in the United States, you'll hear more about that later. And that's, and of course by Kelly's foundation, which was actually sponsoring the reception yesterday. I will first, before I start the introduction, how the syndrome was found, I will first take you a little bit through the program and also show you, give you some background why we think this is relevant. Please realize that also the scientific meeting, I said it yesterday, very predictable for us. We do it every few times a year. This time of meeting is for us also new. So if you have any questions, just raise your hand and ask. Where was the program, Dale? Here? Ah, brilliant. All right, welcome. Okay, the morning session. What's the idea? First, well, originally Nathalie van der A was scheduled. Unfortunately, she's ill, so we replaced her. I'll give you some insight in how this syndrome was discovered. I mean, it is not that old. The second talk will be by Anke Van Dijck. Some of you may know her. She has analyzed a group of patients just to determine, sort of see the common problems between all those children. I assume that many of the children, of your own children, were amongst that study group, so her name may be familiar. Then we have two, of course, we're trying to, the common note of these European network is that we try to do something for the patients. In the morning sessions, there are two talks that we're going to give you some insight in the way we are thinking this should happen. There'll be two talks. Claudio is from our center. He will explain to you how we work on what we're doing on a mouse model. Ludovico, he's from Milan, Italy. He will explain. They're from a lab which is an expert in cellular models. Some of you have donated your fibroblast. This was years ago, and you wonder what happened to them. It's going to be much clearer after Ludovico's talk, I'm sure. Then there'll be a lunch with a little bit more space here. There's coffee break in between. Then we have Patricia van der Wallen. Some of you participate in the gait analysis. You wonder why you haven't heard, but because unfortunately COVID came and then that project suffered so much it was interrupted. But she will show you some intermediate results. We really are doing something with it. Then the other afternoon will be. We're very proud to have Ilana Gozes here. She's Professor Gozes from Tel Aviv, Israel. She will explain you on a NAP trial. I'm sure some of you have heard of it, left, right, and she will put this into a perspective for you. We get much questions on the ketamine trial, clinical trial that's been ongoing, and it's in the Seaver Center in the United States. So we've asked them to come and present. Please realize that for us, it's difficult to comment on it, but they will be here. They'll be the only ones that will not be here. They will present online. They were willing to send someone, got sick, etc. And so, but online, we have a Zoom meeting. We have Dale, who's going to make it work. Then we have Sandra, of course, from the US Kids Foundation. She's really built a society in the United States. Many of you, I think all of you, must have heard from her as to the Facebook group. I think that will be if you want to proceed, that's an instrumental. I think we can benefit very much from. You all can benefit from her experience very much. Although there are of course differences between the United States and Europe, as you all know, and as will become clear. And just to digest it all, we think there'll be with all your questions, you can ask questions directly. Afterwards, there'll be a panel discussion. Basically all the speakers will sit here, and you can fire your questions, and then there's a concluding session on as you wish. Okay. Questions so far? So I'll give you some update on the syndrome now. There it is. How did this all start? I've shown this picture of this figure 100 times, and I would really like to do it another 1,000 times. This all, because actually it's a very peculiar situation. I'm Frank Kooy. I work at the center. Oops, should go Ben. I work at the Center of Medical Genetics. So we're a big center. We do we have a routine part that does the diagnosis of the sort of routine hereditary disorders. And I'm actually at the research part. So what does it mean? I work at the part that if the clinicians say, oh, nothing comes out of the routine, but say, there has to be something, then they will quite often consult us and can you do something? So this is also a point, because this whole story for us started with a young boy. You might recognize him, although he's a little bit older now, with a global developmental delay, failure to thrive, autism spectrum disorder. This may all seem very familiar to you. The mother insisted, said there's something wrong. So she went to Nathalie van der Aam, my colleague, said something wrong, do it. Nathalie is a very knowledgeable clinician, so she came to me, said, Frank, find it. Okay. So I told my students, you have to find it. So the credit goes there. But in fact, it is very important, because what had been done, I guess you all know that, we've done at that time, this is not even 10 years ago, but we've done all the, the chromosomes, nothing there. It looks a little bit like Noonan. Half of you must have heard it. Oh, let's look at this gene, let's look at that gene. It's all negative, nothing. And we were fortunate at that time, and this is maybe a little bit technical, but you don't have to understand the details. At that time, there was a novel technique had been developed. So the sequencing, so determining all the base pairs in the DNA, which was till 10 years ago, you had to do basically on a gene-by-gene basis. Is it this gene? No? Okay, next one, next one. There are 20,000 genes, so this explains why it could take a while. I mean, usually people give up. You could do all genes now at once, but what's normal? We never know. So what the technique that they developed, actually here in Nijmegen, that enabled is that if you don't only sequence, do all the variations in a child, but also look at the father, the mother. That's why we take in. That's why they take at the diagnosis your blood of the parents, and you compare it with the child. You see that all the variations. We inherit one chromosome from the father, one chromosome from the mother. You see that the variations, they're present in the child. If it's present here in the healthy father, possibly not causative. Is it present in the healthy mother? Probably not causative. If it's not present, hey, let's have a look there. So this was the situation, and there was actually, Bert de Vries was instrumental in that. He was here at the scientific meeting yesterday. So then this all seems very easy. But realize that any of these results will give you, I mean, none of us are identical. We're all different, and this is in part is due to the variation in our genes. So if we sequence here, we find about 20,000 variations. That's a lot, if you want to check them. So then you have to filter until you finally find the de novo variants. These days, that's all what we call routine pipelines, the programs for it. We can do it. At that time, it was not. So the bioinformatician, in this case Geert van der Weijer, our bioinformatician, in the course of his PhD, helped developing all those filter steps. And that enabled us here. Here's an interface that we actually built, an interface that we're actually still using today in routine diagnosis. And then we found here, what you see, what did we find in this young boy? A 4 base pair deletion. You can see the bars here, in a gene called ADMP. At that time, that meant nothing to us. We had no clue what it was. If you look, the situation changed since 10 years ago, and that also explains why there are more and more diagnoses, and no one was diagnosed before that time. So we went into the literature, and it wasn't there in the parents. You know, it's as we call a frame shift mutation. So it's really, there's no more functional protein, not possible. You look in all control databases, etc., in all normal controls, you couldn't find it. And then we looked in the literature. Ilana Gozes has written quite a bit on it, and we find that the gene is expressed in brain, that it has a function, that it has been named, involved in neurogenesis, in heart development. There was even a mouse model that gave some problems. So we thought, okay, hey, this is maybe this is interesting, and it could be that if as a person you miss the gene, that it could be related to the disease. But you always have to be, I mean, in research we're always careful. So we never say, this is a problem. We didn't go back to Kelly saying, you know, we find maybe something, you know. We didn't report anything back. So what you do is you go in literature, are more variants described. There was actually a study by O'Rourke and Evan Eichler's lab. They described two variations, but they hadn't sort of, in their study, the frequency was not high enough to draw a conclusion that it was disease-causing. The other genes they called disease-causing. These one they said no. It was sort of to say below that threshold. They worked a little bit different, and it was also, and it confused something in what we thought was a normal control. So what do you do? What did we do? We were all there, so we had the first patient. So you call all your friends, literally, and you meet and you say, hey, do you also have variations in this gene? And we came to Sicily, our collaborators, and of course they had one. They always have a patient there. And then we went to Nijmegen, the Netherlands. First they say, no, we don't have it, but then, I mean, these are clever students always, so they went back into the data and said, no, to the supervisor, you missed this one, it's there. This is not it's true. This was Anneke, it was. And then we went on, then Celine, the student, Celine Helsmoortel, the student, she said, you know, Frank, I'm going to screen. I said, don't waste your time. I mean, this is such a rare disease, don't screen for further patients. And she did, and she found one. She was the lucky one. She found one additional, so as students, yes. My advice is always good, but not in this time. And so it went on. There was a Swedish collaborators. To cut a long story short, then we had the 10. Then we had, in the end, at one stage we had 10 patients. You can see where they were all from. All of them were mutations that stopped that sort of inactivated the gene completely. There's no doubt. There's no function. There was never a doubt. There's no one copy of the gene is missing. And then, in all cases that we knew that we had access to the DNA of the parents, that was not available. They were de novo. So, if you have 10 and then you compare, we compare the clinic, the facial dysmorphia, and you see that there's clear similarity. They're not identical, all of your children, but there are, from a clinical point of view, there are similarities amongst those. And we also do it like you make tables like this, and you see, hey, half of them have this. For instance, short statue is going to be much more details now on this in Anke's talk, where we did a much larger cohort. But these sort of tables enabled us to convince our colleagues, say, hey, now we have proof that this is disease causing. There's some more statistics which were done by Evan Eichler, our partner in America, in Seattle. They have a little bit approach. We work from a clinical point of view. For us, this was enough evidence. For they're more statistical. So they did all sort of statistics on it, and they basically calculated that the chance that this was not disease-causing was like 2 times minus 18. So basically completely ignorable. And they gave us the publication here, and then we submit it to a scientific journal, and then we called it this mutations in this gene are caused by de novo mutations. These are the key characteristics: autism spectrum disorder, intellectual disability, facial dysmorphism, congenital abnormality. Yeah, something about the frequency. This is a very crude estimation based on those 10 patients that we had that time. How did we do it? Well, very simple. We have one patient in a few hundred. So in our case, it was 100%. Other people had one patient in 10,000, so 10 to the 4. We just added all the cohorts and said so many patients is what we found. So this is it's not a final estimation, it's just a crude estimation on the frequency. What about the name? Celine Helsmoortel, she was the PhD student on the project. Nathalie Van Dijck, the clinician, so she's the first, she's the last author. And that's why the OMIM, the website that gives official names to these diseases, I mean, this is driven by them. They call it Helsmoortel Van Dijck syndrome. So one day you open the OMIM and they say, hey, did you see the names? So no, we hadn't seen it. So that's where you can call the disease as you want, but if we published, we're required to use this journal policy enforces us to use official names. Helsmoortel van der A. It doesn't always happen, but nothing is perfect. Same with the gene. Gene is called ADMP. This is the official name. We can say, oh, we don't like the name. We give it another name. Journal won't. You can do it amongst you, but the journals won't allow us to use any other names, just for obvious reasons. Huh? If you go have a passport, you have your name. You call yourself as you wish, but in your passport, that's your official name. It avoids confusing. Great acknowledgement to, oh, it's not a very sharp picture. Who took this? This was actually, first of all, this is I'm standing here in front of a big consortium, Genome Sciences, Evan Eichler, immense Bert's disorders, our two collaborators in Sicily, Karolinska Institute, and there was a patient in Australia. This, all contributed. So we discuss. This is what we're going to work. This is really a group effort. This was the research group at that time. Current people might not even recognize. Say the half-life of a group in research is not that great. Nathalie Van Dijck, she was she was a clinician, clinical geneticist at the center. She still is a pediatrician. She left the center, but she is still around, unfortunately not today. And she was also affiliated with a research group. So she was instrumental. This is Celine Helsmoortel. She did her PhD, moved on to a few other places. She's now with Roche. Not sure where she is actually today. I'm sure I just don't know. We're still in touch occasionally. Gert van der Weijer, the bioinformatician, is more on the background, but he has been very instrumental, sort of this finding, these abnormalities. It would have been found otherwise, but it might have taken another year, 2 years. So at that time, there was really a very great effort. So I think with this, I can end and give the floor to Anke. Unless there are any questions. Sure.
Yes. So you see a lot of differences between. So hello, good morning everyone. I'm Mauritia, I'm a parent. So you see a lot of differences in the kids, right? In terms of development, but also behavioral characteristics. Is that also explainable by the deviations in the genetical code, or where the defect is, or?
Ooh. That's the million-dollar question asked very much in the morning. So the question is basically, is there any relation with the code? I think we better answer that. Anke has done, this was the initial, much more patient, so maybe we should come back on it after the next question. There's some indication that there is, but to what extent? We don't know. But there is clearly some variation, but yes?
Thank you so much. My name is Keith Shannon. My family lives outside of Washington, D.C. One question I have is intellectual disability, the label intellectual disability. What I read was intellectual delays, and then I saw 10 out of 10 have intellectual disabilities. In the U.S., the definitions are different, legally speaking. So I wanted to understand better from your perspective, when you say intellectual disability, how can you describe that, like, more precisely?
Yeah. So first of all, thanks for the question, which is basically a nomenclature issue. When I speak about intellectual disability, to be honest, I'm a molecular biologist, so I'm used to people coming to me with tests and say this enzyme works or it doesn't work, or the mutation is there, so it's not, or it's not. In my case, it's just the doctor gives them a test, and they say if the IQ is below this and this threshold, we call it intellectual disability. So that's how I perceive it. I know there's room for many more nuances. In the early days, you call it developmental delay, of course. So it's a bit of a broad. It's an oversimplified definition, because I'm quite aware that there are gradations there.
How big is the range of that?
Anke's going to explain that. Thanks. Thanks. Please go ahead. Anke's came into the research group actually as a clinician, as a neurologist. She came in when Nathalie left. She did many of the follow-up studies, and I think you know, she did a PhD then later, moved on, but still affiliated and comes back occasionally, not enough, but occasionally, and still finishing. We're still in touch. Thanks, go ahead.
Thank you, Frank. So welcome everybody again, and welcome to the parents and the children. And I first want to start to say, apologize for calling your children patients, because they are just children and we all are parents, and it's because of, please come in, it's because of our background in the hospital. We are so used to naming them patients, but of course they're just who they are. And not everything is a problem or a disease, but the nuance got lost sometimes. So I will take you further into the topic of the clinical spectrum, and there will be some overlap, but that's not bad, I think, because there will be so many information today, and in the evening you will go home and whoa, it's sometimes mind-blowing. So again, who is who, and Frank has shown you already, Nathalie, Céline, and of course also Ilana Gozes. So we could meet several times, and that was very nice to work together, because the syndrome, it's caused by a mutation in the ADMP gene, and that gene plays an important role in brain development, and also many other organ systems in the body are affected when the gene doesn't function well and the protein is not functioning well. And what we saw was there is a syndromic image. It's not just one disorder, it's intellectual disability or a motor delay, speech delay. Also autism is very frequent, and there are several features. I cannot list them in one slide, and also facial features, because in genetics it's common to look at the children and to measure distances between eyes and the nose, and look very carefully. Could we recognize it by watching your child? And we have listed lots of the features. So, and it is prevalent in 0.17% of individuals with autism spectrum disorder, but the number doesn't say much. It all started with that first publication with the 10 children that were described, and Frank already has shown you how the process was. But one of the important things was, can we manage to collect clinical information? Not just knowing that's the gene, or that's the child with the mutation, but even what's the child like? Is it functioning properly at school or not? What's the reason that they went to the doctors? And which kind of evaluation was already done? And that was very difficult. Sometimes we only received the mutation and not many details. It's not common to collect the details on every child with the name and to go back, because sometimes the groups of children were large that were analyzed in the genetic lab. But still, then I came in the lab, Frank Kooy, in 2012, 2013, and we said, wow, we need to go further into it and to look deeper. And we started receiving emails from all over the world, and in the beginning it was weekly, that there was a clinician and that sent us an email, and he or she said, okay, I have a child with a mutation. What can I do? Could you give me some information? And then there were parents emailing us, and then it got weekly and monthly, but we immediately started writing down, okay, there is one in Sweden, there is one in the United States, and 2 in the United States. And then Sandra came, and she, the mother of one of the children at the United States, and she started with an Excel sheet or just write down and collect and share the data. And that was very nice too. From the beginning, we all worked together, and we shared the data and looked, okay, that patient from Sweden, is it the same one that Sandra had on the Facebook group that she at that moment had started? And yes, it was. And then immediately we saw the importance of being part of that, and that was quite new, because as a scientist or as a medical doctor, you're not on Facebook connecting with your patients. That's a little bit weird, but in a few weeks it brought us way further than before, because you could ask directly for missing information. For example, we had information on the development, for example, but we didn't know anything about the teeth or the cardiac status. Was the heart normal? And then we could ask, was there a cardiology consultation? Oh yes, of course. And I have the letter of the clinician. And then immediately I could fill in all the details. So that was very nice working. And in 2018 we could publish the details on 78 participants, which was quite a lot in that time. So just in general, the country of origin, many patients or children came from the United States and also in Europe. Of course, it depended on the centers that we were connected to. It's, of course, when you can ask your friends, it's going much faster than when you don't know them. But yeah, that was in the beginning. And we had more details about male children than female, but it was 50-50 afterwards. And especially younger children, 3 to 12 years old was the mean age, and the eldest one was 40 at that time. So we didn't have a lot information on the elderly, the patients. So here you see the map that I got from a website, and I will come later on that topic on the website. So about the development, many parents told us that their child was sitting at the age of 12 months, while in yellow you see the normal age range. It's about 6 months in the typically developing children. So the white blocks show that a few kids are even sitting later or need help with sitting, how is it called, shelf, to can sit properly. And also the walking age was different from the typically developing children. Normally between somewhere around 1 year of age or 15 months, but you see that the top of the curve is later in the children with Helsmoortel van der Raas syndrome. We also saw that some of the children couldn't walk at all, or maybe they are just too young at the moment of the evaluation. When we saw them at the age of 3, maybe with a lot of physiotherapy and help and aids, they could walk at the age of 10 or 12. There are examples of children that just need that time. So I already said it in this meeting. Actually, we need a new evaluation now. 6, 8 years later, it will be a difference in our data, maybe to know is there an improvement in the walking pattern while getting older. So to come to that point of development, it's indeed so that the clinicians just send us the reports. There is a mild intellectual disability, or when we are lucky, it's just intellectual disability, then we know already something. And then a lot of the child neurologists make the categories visible so that when there is an extensive developmental assessment happened, then they write down there is a mild intellectual disability. The IQ is between 50 and 70, but that's a cutoff. But we tried to add it. There are several children that we don't know about it, or maybe in the countries the tests are a little bit different. So, yeah, the most important thing is that they are delayed in development, and quite a lot, almost half of them are severely delayed. That's important, especially because they need to go to special schools. About the facial features, we just counted them, and we saw that a lot of the children have a high forehead, a prominent forehead, and a high anterior hairline. You see it coming back with lots of them, that the distance between the eyebrows and the hair is quite long. Not in all of them. The nasal bridge, that's the part between the eyes, is often a little bit lower than in the typically developing children. And wider. The nose is rather short, and the tip is often upturned and full. So that are all things that the geneticist is especially for educated in to recognize this. We saw also that in the baby age, the infant age, the chin was rather pointed. In a lot of them, but of course we only have the pictures, and I think I saw at that time 10 of the children. I traveled sometimes, and then, for example, I was following this morphology course in Manchester, and then the Manchester parents asked me, please visit us, and I went by train all there, and it was very nice to see them in real and to know if it's correct or not. Ear malformations, it's the rather variations. The ears, look for a good example, but it's more difficult to see here. The ears are cupped and less developed than in the normal population. Sometimes the earlobes are uplifted, or you see all kinds of variations in the ear, in the form of the ear. General health problems. This is quite an extensive list. What we heard from the parents is that feeding was often difficult. The period in infancy was difficult because of the sucking reflex was less strong than in a normal child. Breastfeeding was difficult. Giving milk, and then there was a failure to thrive. They couldn't grow well in the beginning because they needed to learn how to suck the bottle. A lot of children had reflux disease, also causing a lot of difficulties in the behavior and in understanding why is the child behaving so bad. In 83%. So that was striking. And constipation is also one of the features that we hear a lot, and we cannot completely understand at the moment, but a lot of them are have difficulties, and the challenge is to know, is it just because of for example, the autism, making them eat only the same things, and it's difficult to bring on a diet that is very good to for the digestion, or is it typically for the syndrome? But counting it, it's more than in the population with autism. Visual problems, and also visual problems that are discovered at later age. So hypermetropia, meaning that the children have problems to see from a short distance. Strabism is often reported, and a cortical visual impairment, meaning that there is a problem in the brain interpreting the visual input. Some of the children had MRI scans. This could be because of seizures that the child had. Brain abnormalities were seen, but not specific. So you have, maybe in the reports you see corpus callosum agenesia, or white matter disease, or atrophy, rather aspecific. A lot of musculoskeletal abnormalities, special fingers or toes or feet, or the position of the feet is different. My colleague will come to that later. The heart, we see heart defects in some of the children. For example, ASD, VSD, there is a hole in from the left to the right atrium, for example. Of course, only of the children that we know that the heart is checked, has been checked. A lot of children have ear, nose, or throat problems. We hear lots of airway infections at a younger age. The urogenital malformations are also rather frequent. Sometimes one of the ureters is not developed, or they have lots of urinary tract infections, or there is a problem with one of the kidneys. In a lot of cases, then the child has a problem, and the parents go to the clinician and ask, okay, why has my child so many problems with infections? And then they look further into it. Endocrine abnormalities. There are children with hypothyroidism. The thyroid gland is functioning less. They need medication for that. And also growth hormone problems. Some of the children have a short length, so discovering that, you could go further into that and discover problems in the hormonal balance. And then short stature. in 23% of the children. So a wide variety of features. Here is the test. I went to the ophthalmologist because I'm not educated in ophthalmology. And how can you discover cortical visual impairment? And they showed me a lot of booklets with special silhouette figures. And yeah, it's the whole test battery, but I cannot explain properly. But it's important to check if the vision function is good of your child. Behavioral problems. And I already told that a lot of children have autistic features or the diagnosis of autism spectrum disorder, 93% here in our cohort, and also aggressive behavior or temper tantrums. Sleeping problems are very frequent. Some of the parents use this kind of tent to make the child more comfortable and willing to go to bed, taking away the extensive overload of input that it has in the day. Obsessive compulsive behavior, mood problems, and ADHD features. A lot of children are hyperactive. Which kind of evaluations are needed to be done? A lot of children went to the clinical geneticist. The pediatricians are involved, and we recommend to do a blood test to check the thyroid function and other blood results. The child neurologist is important when you think about seizures or when you see that one side of the child is less developed or can move less. Then maybe they should check with the brain MRI or an EEG. Not telling you that every child needs to have an EEG or a brain MRI. It's always the interpretation of the level of functioning and what the clinician sees that's important. We recommend to go to the cardiologist once just to check if the heart is okay with the sonography and an ECG. Then the ophthalmologist, and a lot of children go already to the physiotherapist, the speech therapist, or the dietitian, and then a psychologist or a psychiatrist is involved in many cases. We had a family day in 2016 because the lab needed more information and more correct information on the children. So in Amsterdam in 2016, we went there and we were happy to see each other and to could ask questions, and it was a good experience. There was a nice cake. And then we also needed patient material, because Frank just told you that he got fibroblasts from you. Of course, you didn't donate fibroblasts. You maybe your child had a skin biopsy, and then they went working on it, and we can do a large amount of tests, and we got a lot of insights by collecting hairs and nails, and that's why on that day also you see the father cutting the nails of the child. It was very nice for us to have this material available and to send this to our collaborators in Canada, in Israel, and to do tests on it. Especially because we already had a lot of information on many of these children. And then coming to the website, one of our colleagues in Nijmegen, he developed a website to collect the information on a more structured base. Our sheets are also fine, of course, just the Excel sheet, or maybe Sandra has already a large table with data and another collection manner, but Bert de Vries from Nijmegen, he had a human disease genes website, and in that website there are many genes. Was it 1,000 or 2,000? I forgot. 1,000, yeah, sorry. And oh yeah. Okay. And he asks us to adopt the gene. So the researchers all over the world that publish on certain genes, he asked them to adopt the gene and to be responsible for that gene. And so we are responsible for the ADMP gene. And we ask clinicians that contact us to fill in the data on their on their patients in this platform. And you can go there, and there is a professional part and a family part. So at first place, they developed the professional part to, as quick as possible, could fill in and learn from the data. And now, yesterday, he told us that by the end of the year, they hope to finish the family part. So clicking here, the clinicians can upload clinical information. So when you know that your clinician is willing to share information and you want to share the information on your child, please ask your clinician to fill out the form on the website or to contact us to know whether or not your child is in the database at the website, because this is long-term work. In 10 years, 20 years, it will be there, and we will work further with this information. And the family part, they hope to give a chance to the parents to fill in other things that you know about your child and in a structured way, to learn on it. And we will see further this day that this brought us more far, for example, with the teeth of the children, but we will come to that later. And then you have these kind of tables. And you can follow this sometimes when you look, and there are more children, and now at the moment 82, and you see, okay, do they have motor delay? Yes, it's a large part, and it's changing by filling in the data. And then there are more numbers, and you can check whether or not there are other children with, for example, spasticity or drooling or such things. So that's interesting. This work couldn't be done without the help of many, many people. Parents and children at first place, and also the research groups, the cognitive genetics group where I belong to, then the ADMP Kids Foundations in Belgium, in the United States, the labs that we're collaborating, and the grants that are given to us. So thank you for your attention, and I will be here all day, so I'm happy to answer questions during the meeting.
Any questions for Anke?
Will you share the slides after the presentation?
Yes, of course. I will leave out the pictures. Sorry? Okay. Well. Yeah, today is recorded apparently. yeah, sorry.
Yeah, so it's recorded, and maybe also Sandra, she can put it on the Facebook page maybe, so it will be shared with everyone around the world so that everyone can have these presentations available. But I thought you would have a question for Anke.
Yes. Thank you. My question is, have there been detected links with skin problems, for example, conditions like piboldism or café au lait spots?
I'm not aware of it. Skin problems is very rare. I think I had one email from a clinician about this, but that was a while ago.
Okay, so Ilana will comment on this further.
So actually we have a paper from 2019, I think, before Corona, with a Spanish group. And the patient is P-tyrosine 719 stop mutation. Actually, they also have a stem cell line from this same patient. And we did see skin problems. The skin is thinner. And we actually verified it in our haploinsufficient mice. And we could treat it with a NAP davunitide. I'm not going to talk about it in my talk, but this paper is out. and you can, it's published in a scientific journal called Scientific Reports, and my name is on it, so you can find it. Thank you.
Okay, one more question, and then we can go on a coffee break.
Thank you so much. May I ask, looking into the future, and I apologize if you've addressed this earlier, what are your plans to continue to try to get more and more information, and how would you project, and how would you expect that to change any of your findings now? Just curious.
Thank you for your question. Of course, at first place, we want to make an improvement for the children and for the parents while growing up. And to do that, we need to know a lot about the syndrome itself, because what are we looking at, and what can we improve? What are the problems? What are the struggles that the parents have and the child has? Behaving and being at school and with siblings, and we need to know that, and then maybe we could do something about it, and there could be a treatment, and we are already trying a few things to correct the mechanism and to see what happens. But of course we also need to know, by doing an intervention, will it mean anything to the child. So in previous clinical trials, for example, we saw that there were children with problems, clinical trials in other genetic diseases, for example, and they gave medication, or they did a non-medical treatment, but we could not measure the effect well. And in order to know what we could measure, we are looking for biomarkers. We are looking for features that we can follow during the time and that we can compare before and after. So that's why we are looking so detailed to the children now. There are some projects that we are doing at the moment. For example, the motor development. We are evaluating the gait features, and we are evaluating the development in general in a group of children, and just thinking what kind of projects we are running at the moment.
There will be more details on the project
in the next presentation. Yes, yes, of course.
Hi, so you talk about it's important to diagnose the cortical vision impairment, but when it's diagnosed, what can you do to improve it? You can improve it by glasses, or is more therapy? Or because I'm sure my son has problem with it, but if it's that the brain cannot interpret the image properly, so I'm okay to diagnose, but if you can do anything about it, what's the point to diagnose it?
At the university hospital, we have a center that is a rehabilitation center for. We have a rehabilitation center for ophthalmologic disease. But unfortunately, I'm not involved in it, so it's a good question. I cannot answer it very well, I'm afraid. So it's important to look for the problems and then to know which kind of training you could give. I don't think it can be corrected by glasses.
Hi, good morning. Our son has glasses, and when he's tired and he doesn't wear his glasses, then his eyes would shut down because the one that is seeing correct will do the work, and that's as we have discovered with our physicians, it's been driven by the brains. that it will shut down. That's something that we heard in the eye hospital in Rotterdam. So yeah, maybe this helps. We have another question for ourselves.
Oh, yeah. I will give it immediately back. What I see with Cedric is he also has the CVI, and when they try to learn him to read, it's important that you just hide the lines behind it, because otherwise then they are focused on everything, and they cannot focus correctly on that one particular line that they want to try to read. So these are all tricks, actually, to overcome this. Yeah, okay. I will just come back to you, but I saw there that he was first with a question. So, okay, thank you.
Just a quick note on CVI. There's a fantastic website called CVI Scotland with one of the utmost researchers on that subject. So I clearly recommend that. And
okay, great. Thank you, Jesper. So CVI Scotland. Need to look it up as well.
Thank you. Yeah, our son is one of the ADMP kids that's very small, and we now just had a prognosis that he will be somewhat a prognosis that he will be around 1 meter 60, 67 or so. Yeah, 67. But we're doubting to do the hormone therapy for him so that he becomes more taller. Is there anything that you can tell us what works, if it works? And I don't know if the next lectures that are coming are more elaborate on this part or not.
Not on the growth hormone, and
okay.
I don't think so. It's important to look with the clinician. Is it good for him to correct the missing hormone or not? Maybe you don't want him to be tall and heavy or sometimes there are lots of considerations that need to be made when to correct or not and what will be the consequences in other organ systems.
Yeah, that's our question.
From the doctor, she asked, she has I make a picture from
all the recommendations.
Yeah, but I know that the children with ADP is faster in hormones. Let's say in puberteit. What is that?
Puberty.
That is they are, but I don't think she knows that he's now 10 and when he stopped.
When's the time that he will stop growing? So they don't know that the estimations are correct for him.
No, our daughter is all longer. She's 8, then he is 10.
Yeah.
So yeah.
Our daughter, she's 15 now, and at the age of 10, they saw that the pubic hormones were stopped. So we try Lucrin injections, and now she's a little bit taller. You have to try it at the hospital. Yeah.
Yeah, and we grow actually after we got here, and he grow in 2 centimeters.
In a half year then, okay.
There are more children having this problem, and I'm sure Sandra can comment on that. Yeah.
This is one of the reasons why it's so important for all of us to share our information, because there are numerous children who are on growth hormones, and we've actually seen improvements in other areas, and they grow until a certain time, and if we collect all that information, we'd be able to create more of an understanding of, when you would stop it, or even what kind of benefits and what kind of negativities are affecting our kids, because like we all know, not all drugs work the right way on our children. So I think it's really important that we all, it's one of the reasons why I'm here today, and I'll talk about it later, but we have a lot of families on it, and growth hormones are one of the things I would love to talk about because it's doing some amazing things for those kids.
I want to add something there in the group, in the Facebook group, there's a lot of questions about growth hormone and other hormones, but it's not only about growing. the body needs growth hormones for building up your muscles, for your bones and everything. So, and also to develop mentally. So, and if most of the time, if the doctor prescribe growth hormone, it's because there's a lack of it. So, you know, it's not like a drug then, but it's a replacement of because of your body is not working. And our son has the same problem, and there is a growth poly for the Dutch parent in the hospital in Amsterdam UMC, and the part is VUMC, but I think now in AMC. So it's a special growth poly, and I think there are more in Holland, but there is one in Amsterdam.
Okay, thank you, Gaby. So I would suggest we go on a coffee break, and then we can even have there some questions. Okay, so it's just out of this auditorium, and then the stairs upstairs, and then there we will find the coffee. Okay, thank you.
Thank you all. As I said welcome back. As I said, sort of in the beginning of the meeting, we will now give you sort of 2 updates on scientific work that we're doing. And this is, and we just realized we're very aware you have many short-term questions. These therapies and this work that we're doing is mainly based on the long-term sort of improvements with a long-term perspective. And Claudio Vincal, he's from Antwerp, Belgium. He's affiliated to 2 groups, primarily with the protein signaling group from Wim van den Berg is the promoter. He's over there. He's also co-organizing this meeting and also affiliated, fortunately, to our cognitive genetics group. And you'll explain now. So please go ahead, Claudio.
Thank you, Frank. First of all, welcome to all the parents. Welcome to the present children. Today I'm going to talk about something that we're studying, and I devote actually a big part of my life to the ADMP gene in mouse models. So, but first, before I want to get started, of course, I want to make a clear statement that anything that I will be saying today, as I'm comparing some mouse features to some features representative in children, that I'm not comparing mice to humans. So please be aware of this. I'm a mouse man, and I really love the ADMP children. So that's the difference. Okay. First of all, it's working. At the university, I can also teach with both of my professors. So I will also give you a little introduction to basic cellular biology, because I think you will need it in order that we can understand each other. You know, our body has millions of cells. And in the cells, we have also organelles. These are the specific organs of a cell. And for example, we have a skeleton. It keeps us standing up. But in the cell, you also have the cytoskeleton. And this is to keep the cell in a nice shape. You also have the core of the cell, and we call this the nucleus. And the nucleus is a very, very important region in the cell, because this is where ADMP mainly is. If we focus here on the nucleus, you see that we have different chromosomes, and you're all familiar with chromosomes, I think. They're like a supercoiled structure, so when we unwind the chromosomes, we get something that we call a nucleosome. And a nucleosome is actually, you can compare it to a phone like the old phones, the supercoil, around some around some circle. And when we zoom in on the region here in between, you get DNA. And in DNA, you have genes. And these genes consist of 4 different letters. And these are G, C, A, and T. Yes. So, very complex figure. Don't be afraid. I'm going to walk with you through it. So this was the core of the cell. And the first thing that the core of the cell is going to tell us is, hey, we have to make something. So we call this process transcription, and the cell is going to message, because it's going to send out a message, because we want to make something. And we call this process translation. So we have received a message now. Now we are going to make the building blocks of the cell. And we call that translation, of course. And this is in the form of amino acids. And I know a lot of men that go to the gym, they take amino acids, so I'm definitely sure that you know what amino acids are. And why? Because amino acids, they form the basic structure of proteins. And proteins, we call them the building blocks of the cell. But what happens when you have a genetic defect? If we have a genetic defect, I have to press really hard. When you have a genetic defect in a gene, for instance, a deletion of a base pair or an addition of a base pair, you can assume that something will go wrong in this process. And a genetic mutation, therefore, can result in a different shape of a building block or complete disappearance of a building block. And therefore, it will cause disease, for example, in the brain. And that brings me to the syndrome that has been introduced in the morning, which is Helsmoortel syndrome or ADMP syndrome. And we have already discussed it extensively, but I'm briefly going to recap everything very shortly, that patients or children with ADMP syndrome, they have a kind of building block in their DNA that has been altered. And this causes premature stop of this messenger. So we can also immediately say that later the building blocks will be a little bit different in the cell. And then why you guys are here, and that's also what I find really important to have parents here, is that actually one of the parents discovered, together in collaboration with, of course, Professor Rosés, that primary tooth eruptions are currently used as a clinical biomarker for the syndrome. And of course, all the patients present with intellectual delay and autism. So let's have a closer look to the ADMP protein. I'm not going to go in too much detail, but what I want you to know is that here is a very special sequence, and we call this structure NAP. And NAP is an octapeptide sequence that has been discovered by Professor Rosés. And it attributes to a neuroprotective function of the protein. And Professor Rosés will elaborate on this a lot more in the afternoon. Then here you have a special domain, and this domain ensures that the protein can go into the core of the cell. And then with this latter domain here, it can interact with DNA. So why am I telling you all of this? Because it's very important that we know the structure of a protein and how the protein looks like, because we are going to study it. And today I will present you our mouse model. Is a mouse model for patients with mutations at the end of the protein, and how we made it, how we characterized it, and what we can conclude about it. So, this is one of the most prevalent mutations that has been reported in ADMP syndrome, and it was also one of the first mutations that has been discovered in, of course, Cedric. And we want to mimic this in a mouse, but therefore we need to have some tools to generate this. And we can, if you know Microsoft Word, we actually did cut and paste with the DNA, and we were able to knock out 14 letters out of the DNA, and then we could mimic the same mutation that has been reported in the children. So we do this in the lab, and then we have a fertilized oocyte, sperm and egg together fused, and we plant this in a mouse. And then the mouse gets pregnant and it gets babies, and then it has a baby with the 14 letters that are missing. And then in the lab, we of course did a validation that it was correct, because you need to cut and paste at the right place. And we could see that it was in a very close proximity of the mutation of Cedric. Okay, so what do the mice look like? So here you see on the left a healthy mouse, and then here you see a mouse with a mutation in the ADMP gene. And you can by eye tell that the mice has a shorter stature, and also there is a significant drop in body weight. And we also see this in some of the patients. And then yesterday I got a really funny question from Cedric. He came to me and said, "Claudio, you made a Cedric mouse, can I have this as a pet?" And I told him, "Cedric, no, I cannot do this, because it's a very special mouse, I need it." And then he said, okay, then you can keep it. So but what I want you to tell about the mice is I'm studying the brain of the mice predominantly, and we could detect a decrease in the level of ADMP in the brain. However, when we are looking for the defect protein that I told you about, currently we could not detect it. We cannot see it. And we see the wrong messenger, so the core of the cell is telling us, hey, I'm gonna make this wrong messenger, but the end product, we cannot detect it yet. And that's currently a struggle that we're facing, and it's also something that I'm deeply investigating. Yeah. So next, we ask ourselves the question, okay, we have this mutation in the ADMP gene. How does this affect the anatomy, the brain shape? Because we know that some children have alterations in their brains. And we could identify 4 brain regions that were reduced in size. Okay? I'm not going to bother you with all these difficult names, but the brain regions that we have identified are involved in voluntary movements, social functioning, which is a key aspect of autism that is impaired. Also here, very important, we have a region, and it's part of the hippocampus region, and MRI scans of children with ADMP syndrome has discovered that children have hypoplasticity of the hippocampal region, and we see the same thing in our mouse. And then lastly, the cerebellum is affected, and that's a region of the brain that's really important in motor coordination and walking. And some patients have this typical gait walking pattern. So, okay, it's really hard to compare behavior in mice and men, of course, but we subjected our mouse model to a bunch of behavioral tests that might give us an indication about patient-related features. Again, I'm not comparing, but it's a fun test, actually. What do? We take a platform like this, and we pour water into it, and we drop some paint that the mice cannot, the water is opaque. We let our mouse, after a training period, we let it swim, and we wanted to find a hidden platform underneath the water. And then after a period of learning, we take the platform away, and then we let the mouse reach the same point. And we can measure this. And then we see that ADMP mice, they are slower in swimming, and they also take a longer path to reach to the hidden point. So this indicates that our mouse model has some cognitive impairments. The second test that we did is also an elevated plus maze test. And in this test, normally in a mouse natural behavior, it resides here in the closed arms because it's a safe environment. But what the normal mice do, they're going to explore. They will explore all the things in the maze. The ADMP mutant mice, they don't like this. They spend less times in the open arms. And for a scientist, this is an indication that there are some anxiety-like behaviors going on. I did not put it in the slide, but we did also a marble burying test. And this is a test where we put marbles, in Dutch knikkers, in a cage. And then we see that the mutant mice, they like to hide these marbles. And this is an indication of repetitive behavior, which is a key feature of autism. So I said I really like molecules, really a lot. And I also did a lot of molecular work on the brain, and I'm going to keep it really easy. But I really want to tell you guys this. I said there was something wrong with the messenger that came out of the core. So we are investigating what is going on there. And we have actually a technique in the lab by which we can determine all the letters of this wrong messenger and all the effects on all the other genes. And that's what we did in this part. And we were able to see that a mutation in ADMP caused a massive disruption in gene regulation. And you might wonder now, okay, but what does this mean for the cell? Which processes are affected? And then you could see very clearly that we have, for example, alterations of skin. We also have receptors in the brain that are altered, and they function normally for good action potentials in the nerve cells, and also cytoskeletal dysfunctions. And what's very, very nice is that you already find this in another of your mouse models. So it is actually a good validation that we find this also in different ADMP mouse models. As scientists, we ask ourselves the question then, what is all these genes, what are they doing? How can we group them? We can group them here, and then we see that there is a very important pathway that is heavily down, and we call this pathway the Wnt signaling pathway. And I currently see a lot of eyes going, yeah, what does this mean? I have no clue. Well, Wnt signaling is actually a very important pathway, and when you have normal Wnt, you have this baby neuron-like cell and becomes a mature neuron, a brain cell. When we have a little bit less of this Wnt signaling, you see that we don't form a nice and pretty neuron. And in fact, we also don't make these great connections with each other. And that's something that is impaired in the brain. And I'm sure of it that Professor Rosès in the afternoon will explain a lot more about this. But what was also really striking, the gene that we found that was majorly downregulated is Wnt10A. And this is a gene that is involved in proper bone formation of the jaw, for example, but also for teething. And if I can correlate back to the first slide, we know that the children have problems with their teeth. And here we have molecular proof of that. Yeah, okay. This is something really difficult. I'm not going to go into detail, but we also investigated the building blocks of the cells, the proteins. And when we investigated these building blocks, we could also see that there are some differences. And a major difference is that we could identify here in the center. This is MAPT, it's tau. It's a very important protein in the onset of Alzheimer's disease. And we see that it has a connection with various players of autophagy, the cleaning mechanism of the cell, and also with a Wnt signaling member, which is here. So what does this actually mean? That we have some molecular indication that there is accelerated aging going on. And that's something that Professor Rosès, she found it, so she's the expert on this. But we are really lacking clinical proof of this. So we have some clinical proof, of course, but from living children, we don't know actually how they will proceed when they go onto this late time of age. And that's something that we really want to know, and we can study it now in our mouse model to make some predictions, of course. Okay, so what did we actually find? We find that we have in our novel mouse model mimicking a C-terminal patient mutation, some reduction in ADMP levels, and we have molecular proof, behavioral proof, and neuroanatomical proof of features that we also find in patients. So, I'm briefly going to mention something very, very hot because Professor Gauzès and our lab, we teamed up, of course, because now we have 3 mouse models, and you all know what we're going for. We're going to compare all these 3 mouse models because they completely cover a spectrum of ADMP children. Yes, here. So here you see a haploinsufficient mouse model that is from the Gauzès lab, was the first mouse model. Then recently, Professor Gauzès has made a second mice model, the tyrosine 718 mouse model, which is located in this really important region here. And then what I just presented is the mouse model here at the end. But first, when we go into this, sorry. Oh, yeah. You all know that patients is also on the ADMP Kids website. Patients of ADMP can be divided into subgroups. And that's based on something very specific. We call this methylation. And this is actually a nice modification on top of the letters of the DNA. And we see that when the children have a mutation at the beginning or at the end of the protein, they have more genes that are switched on. However, when the patient or the children have a mutation on the mid part of the protein, we see that there are a lot more genes that are switched off. So this means that there are differences amongst the children, and we need to investigate this. And that brings me to my scientific question: can we differentiate between the mouse models? And I'm gonna make you very happy, of course. Sorry. But what I wanted to say is also this prevalent mutation, the tyrosine 719 mutation, herefore Anke has already made or predicted a genotype-phenotype correlation, because children with this mutation, they have a higher pain threshold. They are also a little bit later in terms of walking. And then you see this really difficult term, and this term just means that they have an underdevelopment of the upper eyelid. So what did we do? This was all work that was conducted by these two Breen and Bent, 2 scientists, and they found this signature specifically in blood of patients. But you know that ADMP syndrome is also a neurological disorder. So can we find the same signature in human blood also in the brain of mice? You know what I mean? It means that if we find this is really, really saying that it's highly conserved, and it's also an indication, if we can find this, that the mouse models are valid. And of course, with help of my great interns, we were able to find this signature also in the mouse model. So you see the mouse model all the way to the left and the right. They show a lot of blue here. So this means that more genes are turned on. And you see that the recent mouse model of Professor Gauzès here shows a different pattern. So that means that also in the brain, in mice, we can make a distinction. And that is really important if we're going to talk about therapy, because that means that patients are or will be treated differently. And we have to take that into account. And then of course you see here this difficult wing signaling coming up, and it's shared amongst the 2 extreme mouse models. And in the middle you see that we have totally different names, so that also means that there is molecular proof that the genes in the brain are also doing something different. Okay. Yeah, and now is it going faster? Yeah. So what I want to conclude about this is really, really recent work. I only did this last month, and I'm working very hard on it. is that we found methylation, it's same signature in blood of patients, and we could track this back into the brain of mice. This is a really, really fascinating observation. And this opens new windows for therapy, of course. So I will go back to the first slide, and I asked 2 questions. I asked, what have we learned from these mouse models? And I hope that I have convinced you that these mouse models are mimicking, in part, the symptoms that we could detect in patients. And now that we have all these mouse models, we can use them to screen drugs. Can we use it for drug screening? Yes, of course, and we have a lot of talks about this in the afternoon, how we can use this thing for drug screening. But what I wanted to say is, we have different genes, and they contribute to different things that are going wrong in the cell. So maybe it's also necessary in the future, and I discussed this also with Professor José and Sandra, that maybe we can also try different things to target different things in the cell, because we have proof for this. And then lastly, I want to share something personal, and I think it's also especially for you. I'm a really, really, really driven scientist, and you guys, you make it really, really nice for me to go to work every day, and for me, this is really part of my life. I really like to work on this, and I also wanted to say I like it so much that the university gave me and the professor the ability also to make a course about this. In the Biomedical Sciences Masters, I teach a course, and it's called Molecular Analysis of Disease. And then we use this nice example of this mouse model to teach the students how to analyze this difficult molecular stuff. So as a university, we want to also shape a little bit awareness for the disorder, for the syndrome, and I'm quite pushy, so I really want this. And then to end the talk, and I would like to thank my professors. I would like to thank Professor José especially. I would like to thank my interns that make my life a little bit less hard. And I really would like to thank you, because you are one of the driving forces to get me to work every day.
Questions for Claudio? Thank you very much for your nice talk, and really, you even got me emotional, to be honest. So, questions? And you were scared you wouldn't be very clear on what you were bringing, so...
Did I explain it clearly?
Yes. Okay, a question upstairs.
Yes, what I keep wondering about is what is the link between Alzheimer's and ADMP? I hear that coming back in more speeches that I've heard, but I really don't understand the link.
So, I'm not the Alzheimer's expert. That's Professor José here in front, so she will elaborate on this more. But we found in ADMP brains this protein called tau, and this is, when you have Alzheimer's, you have actually, the protein is a little bit more, how can I say, it has more light switches, if you understand, and this is getting to the onset of Alzheimer's disease. And we found this protein also in brains of ADMP, a deceased child of ADMP, and also in the mice. So this means that you could have accelerated aging. And that is something that we are very hardly investigating, because if this is true, it means that the children will age a little bit faster. So I think as a parent, you also want to know about the future of your child. So we really, really, really need to investigate this and get to the bottom of this, and we can make predictions about this. But yeah, of course, a mouse is not a human, but we can inform you about some possible things, and that's why it's so important.
Shall I say? So first of all, you gave a fantastic talk, really fantastic talk, and I've mentored many students, but you're something really good. And to your questions, I will actually show some data, some pictures during my talk in the afternoon but Claudio put it really correctly. So this is, I mean, this is like the bottom line. I'll show some pictures and I will be open to answer any questions that you will have. So thank you. But I want also to say something scientifically, that it is very gratifying, you know, when you work on something and your student brings you results. So, you know, as a university professor, I actually tell the students, and they tell me what they would like to do, and they do their work, and they come with the work. And it's really gratifying to see that the work come together so beautifully, and that the results are true and real. And you know, we sent Claudio just brain samples, and he came up with exactly the same pictures that we see in the humans. And the mouse models have so many similarities. So it means that the work is really good and accurate. So thank you very much.
Thank you. Question for you, and then I will come upstairs.
You were showing this slide where you divided the gene into 3 parts, where you were saying the first part and last part were showing proteins. I think proteins were turned on.
Yeah.
At least some things were turned on.
Yeah.
and the middle part was less turned on. How can you know where your child is, like the first part, last part, or middle?
If you know the mutation of your child, I can point this out for you.
Right.
So
I guess
if you have the number, we can also send you the figure, and if you have the number, you can ask us on your doctor or clinician. Then you can actually look for the number yourself.
Okay, cool. Thanks.
Yeah. But you can also ask me to look for you, of course. Yeah. However, I do have to say that not all patient mutations have been covered. That's a little side note, only the most prevalent ones.
Have you started some medication to what you notice, for example, for the excitatory glutamatergic synapse and the methylation problem or the B-catenin? There exist things on the market now, so do you try it on the mouse? Or?
I have just purchased them. So
Okay.
it took a long time to.
Okay.
Now I'm only showing a few slides, of course, but if you will read the paper in December, because then we're going to put it out, then you will see that there is a lot more. I worked really day and night on this, but now the model is valid, and now we're exploring other things for drug medication, and we have great things upcoming. So I hope next time we see each other that I can show you a little bit of those results. But I also have been testing something, and it looks that it's going into a certain direction, but I cannot say anything more about it.
Okay. Because we notice for
Yeah.
we notice for our son.
I'm not allowed.
We use N-acetylcysteine, for example, precursor of the glutathione, and we notice some change of our son, and also all the food that gives a big glutamatergic reaction. When we remove it, it was better, so we clearly see something with the glutamate.
I can also say that because I didn't show the data yet because it's in the lab, but we also see that there are gastrointestinal problems in our mouse, which is frequently reported amongst the children. And we are investigating also, and Professor Gozes also did this in her model, which are the alterations in bacterial flora.
Okay.
So we are not only exploring drugs, but you could also work up, you know, the microbiota
yes,
that are
we
changed.
did a fecal transplantation last year, 1 year ago, and I have the result before and after, if you want me to send you
Yes,
that.
please. But I also wanted to say that besides drugs, sometimes some dietary changes, which are not as, how can I say, intervening, they can also maybe make a difference. And we also should explore these easy things before going into hard drugs, because we're still talking about kids, of course.
Okay, so one more question here from Stefan.
Not a question, more a hint. Even if you can't talk about your further plans, I would be interested in maybe when you investigated, we had some pretty interesting results with a medication called Stratera. It's neurological medication for ADS patients. And if you could try that on mice, maybe that could have some interesting effects.
Yeah, we can definitely try that. The only thing that has to be checked before is if the molecule is capable of crossing the barrier between the blood and the brain. That's really important. But if you say that it's already reported in, I think, AS patients, you say?
ADS and
then
ADHD.
it's for sure the case, and then we can easily test this.
The question was, could probiotics help?
If we're, in fact, yes, they can help, because if you see these changes in the microbiota, you can supplement the bad bacteria with better ones, and then you could have a benefit. But we still have to test this. But if you, we can try it already now.
And also like some supplements as glutamate or that kind of things?
Yeah, these small molecules are actually precursor molecules of bigger molecules in the brain. So if there would be a lack of it, then this would act as a substitute. But I also said to Sandra, therefore we need to have blood of children, and then we can measure this.
Because of the problems of behavior, etc. Maybe there's a key.
Yeah, that's possible.
Yeah.
We know that the glutamatergic synapse is affected, so you hear the word glutamate, so there has to be something.
Okay, thank you, Claudio.
Thanks, Claudio. So we have Claudio, who's from here. It's now my pleasure to announce Ludovico. He's from Milan. This is a group that we've been working with for as long as, basically just after we started working in ADMP. I catched up with his supervisor, Giuseppe Testa, and well, he gave us Ludovico. He was there involved from the beginning, and he will talk about their expertise, because to give us some background, the group in Milan is the group with the most expertise in working with cellular models. Again, some of you gave your fibroblasts to donate your fibroblast. Maybe you weren't even aware of them. They're still alive, some of the cells, and Ludovico does his tricks with it. So please go ahead, Ludovico.
Yeah, thank you. First of all, I want to thank all the organizers of this great event for bringing together different kind of people from scientists. There was a nice exchange in the last 2 days, and now it's even more valuable now in front of all the community. So I will try to explain what we're doing in the lab and what is the main focus of our attention is the brain, and in particular is the human brain. It's very good that Claudio already gave all the basic information about genomics, let's say, and what happens in the nucleus, what happens in the cells, what is the identity of the cell. So thank you. So now we are on the same page, I guess. And it's our main interest it's the brain, which is arguably the most complex organ in the human organism. And to complicate things even more, we want to look at the cortex, which is, again, arguably the most complex subregions of the brain, where reside all the higher cognitive functions, which are often disrupted in neurodevelopmental disorders such as Helsmoortel syndrome. Just to give a brief overview of how complex this can get, these cells that compose the cortex have to migrate, have to differentiate, have to separate from each other or intertwine with each other, and try to make the right connections at the right time in specific areas. And this all happens in a very complex timeline that is regulated by many factors that we're studying as different labs and consortium that Frank was also referring to, which are found throughout all the brain. What we are mainly interested in is this part here of blue of the cortex. And so all these genes have to converge to make these all complex regions to function. What we know about this is that on a spatial temporal point of view, we have to look at what happens at the early stages of development, and this is what we're interested in, because we know that most of these genes, and ADMP among those, it's absolutely relevant for the early stages of the development to make all this machinery function properly. So this is the publication that you've probably seen, the foundation of this work by Frank and Nathalie Van Dijck and Celine Helsmoortel. So this is what we started, and when our collaboration started. And again, with the time, this became even more clear that ADMP was involved in the most important functions to make the brain what it really is. And along with other genes, you can see that ADMP is scored as one of the most enriched genes that is involved in autism spectrum disorder. And the genetic architecture behind it is very interesting to us, so we can try to leverage it in order to study this in vitro in the lab. So what are our objectives? We want to dissect the human ADMP function. So what Claudia has been brilliantly talk about is the mouse model. And we're trying to now see the other face of the coin, which is what happens in human. And to do that, I'm gonna explain how we do that on a cellular model. So investigate the human ADMP function in healthy and diseased states, try to understand what happens when ADMP is mutated and how these cells evolve during their lifespan. And also we want to know, because ADMP and because we know that this syndrome has a vast, not all the symptoms are reside in the brain, but the one that we're interested in, and maybe also the one that you're also interested in, reside in the brain. We want to assess ADMP-related vulnerabilities in a good example of the development of the brain, and hopefully try to connect all these dots. So where do we start studying this condition? So probably most of you knows that we need the substrate to study this pathology, and the substrate is a skin biopsy. So it all starts with this, namely a skin biopsy from the children, and we take that from the children, but also from the control. So we always want to compare what happens in a developmental deficit compared to what is considered a healthy control. However, this skin biopsy is not really what we can use in a lab as a main model. First, because it's not so relevant for the study of this specific pathology. I mean, we've seen also, Anke gave an overview of what are the different deficits that occur in these children. But skin is not exactly what we want to focus on. Also, this is not a model that is very suitable for long-term culture in vitro. This is a technical point, but helps you understand that the skin, again, it's not the most relevant example here. And also, skin biopsy is what we call a terminally differentiated cell line. It means that the skin biopsy will stay skin biopsy. So we can't really modulate that cells as we want to, and I'll show you now how we want to modulate these cells. So in order to understand this, it's good to have a bit of a knowledge of what we I've studied, and it's now part of the foundation of the biology, and especially in the epigenetic field, which is the stem cells and the concept about the properties of the stem cells. So I told you that the skin is not the most suitable model for us, so we want to understand what instead we can do with the stem cells, and in particular with the pluripotent stem cells. So the concept that is really the most famous concept about the pluripotent stem cells so far is that one stem cells can differentiate into differentiated examples of different cells in different tissues. This is a process called differentiation, where the pluripotent stem cells become something different along development. But almost around a decade ago, there was a transformative example of what we can do with these differentiated cells, which means that we can start from them and then go back, let's say, in time to revert their identity back to the stem cells in a process that is called reprogramming. So this is where we do the trick. This is what we actually do to convert the skin biopsy that I told you it's not the best in our hand, to then obtain the pluripotent stem cells through this reprogramming process that I told you. It's a chemical, it's really a chemical process, and it takes little time and not so much handwork to do that. But why are these pluripotent stem cells so important? Because they are representative of the embryonic patient-specific cell behavior. So this can, taken from the patient and compared to the control, we can really try to understand what the behavior of these cells is informing us about the embryonic stage of the development. It's also suitable for long-term culture in vitro, meaning that we can keep, theoretically, these cells in culture forever. It's very good for us to have these in the lab so that we can, that we are sure that we will have these cells in time. But probably the most important thing that we consider these cells useful for is having the ability to differentiate into every organism tissue. So I told you the differentiation process, it can take different time according to different tissues, and as I told you at the beginning, we are interested in the neuronal tissue, and I'll go to that later in a moment. So, but right now we can ask questions about already the iPSCs, because we said that they are representative of the embryonic-like state, which is interesting to us, because we said that most of the, some of the origins of the pathology can already be present at the embryonic state. So what can we do with these cells? I just want to give you an example of what we know now from the stem cells. For example, we can ask, where does ADMP on a molecular level go? So, and where does it act? Why is that important? Because as Claudio said also in his introduction, in the DNA, we can find many different spaces and domains, let's say, that are relevant for different functions. And understanding how a protein can modulate these functions, it's very important to us to understand for basically the first time what human ADMP is doing on a molecular level. So we can start from the genome. Here you can see the chromosomes, and we can try to understand where does ADMP goes. So this is ADMP protein, and I mapped here the mutation that spans throughout all the functional domain of this protein. So the patients that we have in our cohort, and we can try to understand where this protein goes in the broad map of the genome, and this will inform us. And just to give you an example of this, we now know that when ADMP is properly functioning, ADMP is present at the genome, and it makes sure that some genes are turned off while these cells go towards the neuronal lineage. So this means that ADMP is repressing, for example, some genes that shouldn't be there. And it's good, it's a control mechanism, and our cells are full of those control mechanisms, which is good. This means that these cells will properly become neurons. So what happens when ADMP is missing, though? When ADMP is missing, this protein complex is no longer formed. This means that now the genes that were shut down and that they were correctly shut down are no longer shut down. They are turned on. They are expressed. And because the expression of genes encode the identity of the cells, the process that we talked about, namely the pluripotent stem cells becoming the neurons, it's impaired. And these cells has been showed, and we are showing, of course, that the pluripotent stem cells won't become proper neurons. These neurons are not properly formed. They cannot connect very well. They undergo cell death. They are not healthy. So at this point, we can clearly say that we have in our hand a system which is pluripotent stem cells, and we want to arrive to understand what happens in a neuronal lineage. So we can sum these parts as an investigation of the function of ADMP in the genomic regulation, which is the example that I just gave, during neurogenesis. So one could say, okay, why don't we just look at the brain? Why don't we, go and see what happens in the brain? But of course, as you know, the brain is problematic in that is an inaccessible tissue, meaning that we can't physically open the skull of patients and look at the brain. And also, even if we could, in some examples, like in the post-mortem samples, they won't recapitulate the developmental trajectory that goes from the pluripotent stem cells to the neurons. Because you would only have the end point, which is the post-mortem brain. So I told you before that from the skin of these individuals, we can reprogram and have pluripotent stem cells. And I told you the most important properties here. And I want you to focus on the third one, which is the ability to differentiate into every organism tissue. So with these cells, what we are doing in the lab currently and routinely in all our projects is the ability, leveraging the ability of these cells to differentiate into neuronal tissue, in particular cortical tissue. So with these tools in our hand, we can investigate all the developmental trajectory that goes from the pluripotent stem cells towards the neurons. But as the technology in cellular models advances, we can do something that is now considered even more relevant and transformative in trying to address these disease modeling questions, which is taking these cells and transformed into what is called cortical organoids. So these cortical organoids are structures that we can now use in vitro for a number of purposes, and we're now using that in this project focused on ADMP as a disease modeling tool. But what they really are 3D structures that can resemble the properties of the human brain. So, of course, we collaborate for a reason with Frank and his team, with Ilana and his team, and Christopher and his team, basically because the mouse model that, for example, Claudio explained so well, it's informative on what happens in humans, but unfortunately, we all know that the limits of this is that the neurodevelopmental trajectory of the mouse is qualitatively and quantitatively different from that of the human. You can see also summarized here. So the efforts that I just told you about in the field of cellular models is trying to understand what can we improve to better recapitulate these features. And cortical organoids are now quite established as a model that can better recapitulate the developing human brain. This, of course, doesn't mean that mouse is not useful anymore and we can replace it, because the in vivo studies are, of course, very, very relevant. While on the organoids, we can't talk about in vivo study. We're still talking about in vitro. However, this is something that has been evolved with time. So in order to arrive here, we have studied cellular models that are 2D neurons, which means neurons in dish. They can be structured differently, and these are models, cellular models that have been employed in the last decades for many, many purposes. But now we really want to try to switch to this paradigm here, which is the organoid. And as you can see, the complexity of the brain features is increasing while we move from these structures to the organoid structures, which means that it's more hard to study these models, but it's also more informative on different aspects. And I just want to give you an example of what we're doing with these cortical organoids. So they are structures that are chemically patterned to induce the pluripotent stem cells to produce over time structures that resemble the human cortex, which is what we're interested in studying in the case of ADMP syndrome. And when we did this in the control lines and in the patient lines that I showed you before, we could immediately see that there's something going on that makes them quite different in terms of healthy, let's say. So we can measure this, and we can measure this in terms of sizes of these organoids, which means, as you can see, the blue representing the controls and the orange representing the patients, that the size of these cortical structures in vitro are always lower when we study them over time during their differentiation. We can also measure some parameters of their health status, and again, you can see that the controls score better compared to the patient lines, which means that we are gathering hints of what could be going wrong in the development of these structures, at least. If we push this also, and we try this also in many different ways and using different protocols, we can see that, again, the controls that we are using compared to the patients are behaving differently. And again, when we go on with the differentiation, you can see that the control organoids look healthier than the HV does organoids, which show this aberrant cell death, increase of cell death, and kind of abnormally structured organoids. This is something that we can go and study at different time points of this process of differentiation. And of course, we are also interested in characterizing, because we want to assess that the model is good. And the model is good, apparently, because we can retrieve by immunofluorescence, which is a technique of looking at these organoids and try to pinpoint what cells are present in these structures. We can see that both in the controls and in the patient organoids, there are the cells that we expect to be present. However, they are in different structures and in different abundance. So we, for example, can measure, again, the area of these organoids, so the size of these organoids, and you can see that the volume of the structures in the HV-DAS individuals is actually lowered. And if we want to try to understand why they are smaller in size, we can ask how are these cells composing these structures behaving? And for example, if we look at the cells that are neuronal precursors, meaning the cells that populate these structures in order to make them proliferating, we can see that these the patient organoids are more depleted of a pool of cells that is dividing, making this organoid, let's say, less dynamic in a sense. The cells that should be dividing in these organoids are really not dividing. at this point, we can also apply some of the, let's say, cutting-edge technique in genomics field, which is looking at the single cell of this organoid. So, because I told you that the pluripotent stem cells, we can drive these cells all the way to the cortical structures. We don't, and I told you at the beginning that the cortex is also composed by many different cells that have to intertwine and have to collaborate together. We expect to see different cell types in these structures. And indeed, when we can quantify these individual cells in the organoids, and we can see that in the controls, they are, when we perform this genomics experiment at this stage of development, we would expect something that looks like this. Meaning that the abundance of the progenitor cells here, so the cells that are going to become neurons but are still not neurons, is the most abundant cell type you can see in this proportion. While in the organoid of coming from the HVDAS individuals, we can see that this proportion is altered. It's quite altered, and is altered in a certain direction, so we can pinpoint in which direction these cells are going. And the directions are going is an accelerated maturations, meaning that there are less progenitors, but there are more cells that are in an advanced state of differentiation. So these cells seem to be running faster in their developmental path. So just to, I'll conclude with this, and just to give what we can consider a future perspective, we can try to identify some actionable targets among the ADMP sensitive effectors, meaning that when we will have a clearer map of where ADMP is targeting and what ADMP is targeting, we can try to understand if this gene, particularly, if one gene in particular is deregulated, meaning that the cell identity can be compromised, can we restore its function and ameliorate the phenotype that we score? Or even more on concrete side, the drug intervention, can we use it on selective pathways that are intervening in the neuronal differentiation? So once we identify, for example, a neuronal phenotype, an abnormal chain of molecular events, can we modulate it to activate it on repressive and restore that, for example, the relative abundance of cell population that I showed you in the bars before? So these are questions that, as Frank was also correctly saying, are long-term ones, of course, as you understand. But just to give you an example of what we are doing in the lab, it's unfortunately not in the ADMP case, but I just want to leave you with an example of what we have done in another developmental pathology that we're studying, which is these weird numbers that you don't need to remember, but it's again another condition that is affecting the neurodevelopment. In these conditions, there is one gene which is called GTF2I, like ADMP in Helsmoortel Van Terra syndrome, which is aberrantly upregulated, meaning that these cells produce a lot more of these genes than they should. So what we're trying to do in the lab is decrease this abundance of this gene product through some drugs. And we can, in fact, down-regulate the expression of GTF2I, meaning that we can try to push back the levels of this protein to the healthy status of this gene level. And we are showing in this here both on the transcriptional levels, as Claudio was saying, so when the information arrives, and then also on the protein level, which is the final product of this messenger, and that is what we really can score then as an endpoint. So with this, I'll just conclude, and I'll leave space to any question, whatever you are interested in, please ask. And I just want to thank Giuseppe Testa, he's the supervising of this work, all the lab that collaborates to many of these projects, and of course the collaborators of consortium, Frank and Christopher and Ilana in particular. And of course, thank you all for listening. And I think, as Claudio said as well, it's really meaningful to us to then step out of the lab and be here in front of you, trying to, make a difference in this. So thank you.
Thank you so much, Ludovico, for your interesting talk. Questions for Ludovico? Yes.
Yes, thank you very much. Sorry, this is short. This is Mauritia again. I'm very grateful, first of all, for all the research done. This is really great for us to hear. I have one question. I'm not sure if you're really like the expert, but I had one weird thing during my pregnancy, but no one could answer me. So during the pregnancy, I felt in my belly like this trembling. And yeah, it was not the hiccups, because my gynecologist told me it's the hiccups, but it was definitely not the hiccups. And I tried to figure online what it could be, and I thought maybe it has to do something with the nerve system. But that's the only thing I could figure out myself. I'm not sure if you know anything about it, because of course there's this pre-development. It was really like this trembling thing. It was the only weird thing I had during my pregnancy. For the rest, everything was normal. Even the birth weight was somewhat normal, but this was peculiar to me. So I'm not sure, first of all, if it's something you know or something helpful, but it was really the only weird thing.
I am no physician, I am a molecular biologist, so I can't really relate on the clinical phenotypes that I guess many of you are questioning about. But I mean, this is something that could be, for example, discussed, of course, with a physician of reference, but also that maybe we can try to understand a little bit more in detail with Anke, that was scoring all these phenotypes and pre-birth and post-birth phenotypes. But again, the neuronal phenotype that can be, it wouldn't be, let's say, on such a high scale kind of symptoms. Let's say it's something that is relating with the development of these embryos. And for now, we don't have any reason to believe that this will affect overall the pregnancy, for example. But it might as well be the case that in the future there could be different observation on that.
So, thank you for the nice talk. I'm not a clinician, you know. I am a mouse doctor, but we do see like the neuromuscular junction. So the muscle nerve connection is also disrupted, and we actually have another model that I'm not going to show, and I didn't show even yesterday, even to the scientists, where we just knocked down ADNP in the muscle, and we saw a phenotype that was problematic, and which was corrected, of course, by our drug candidate, navtemadine. But what I wanted to say, like what could happen, I think that some of the children, like just from talking to the parents, have muscle spasms sometimes, and there are problems with the muscle development, and that's why some children start walking late, and maybe your child had some spasms, and this is what you felt. I mean, again, I'm not a clinician, and I didn't see this in the mice, but this is just like common sense thinking about it. It could be something that is related to that, and we think that the mutation might affect from the beginning of pregnancy, and the brain, we actually, our original studies many years ago, we showed that ADNP peaks in terms of expression during embryonic development, so this could be associated with that. Maybe.
Yes.
Yeah. Thank you.
So you were first, then Sandra, and then I thought there was a question as well.
Well, what kind of drugs are you going to testing on the tissue? For example.
So right now we are not. Right now, in these models that I showed, we are not testing a specific drug for ADNP. because I mean there are some clinical trials, of course, you're aware of, but at the moment in these structures we are really trying to understand actually what happens in the cells on the pathological phenotype per se. So how to restore it is a second step. So unless we understand until we understand what is really going on wrong in these cells that are populating these structures, you can't really give a specific drug. So what I showed you now, it's probably the tip of the iceberg that we are now evaluating in the structure. But as I said, in the future perspective, the moment we understand the molecular chains of event, then that's where we can act. But at the moment, it's a bit too early on this.
It's taking years, probably.
It's taking. Well, yes, it's taking years. I'm working on this project for 5 years now, and so you can understand how long it takes to build all this. A lot of work has been done, and I mean we are on a way to publish soon our work, but it's still trying to understand what goes wrong in this trajectory of development. But to pinpoint exactly something that can be druggable, that's a different step, and I'm afraid it will require some other years at least to study in cortical organoids. Thank you.
My question was somewhat similar. I was going to ask, how long did it take you to get to the point to where you could do the high throughput?
Sorry, I can't.
How long did it take you to get to the point to do high throughput screening in GTF21?
That project as well for GTF2I, it took also, if I'm not mistaken, 4 or 5 years.
And then you were able to do high throughput screening?
Yes, this is, as you could see, there were 3 drugs written in that could restore the proper level of that gene. And I think in the lab we screened thousands of small molecular compounds, and there were people in a team that were working on it, screening every day thousands of those, trying to make an endpoint, meaning understand what is good for and what is bad for a specific compound, then move on to the other, then move on to the other, then move on to the other,
and then it goes to
takes a lot of time, of course. Yes.
So if we've already done high throughput screening, we have some hits. Can you help us?
If
we've already done high throughput screenings and found small molecules, can your
organoids
be used right now?
Those small molecules are working in that model. It's a different pathology, of course. It's a different condition, but those are working and are working on a specific, molecular chain of events that are specific to that pathology. But those ones won't apply to ADMP. So we can't just take those ones and translate to ADMP. So as for now, it's, let's say, it would be a step into the void, just take those drugs and randomly put into the cortical organoids of ADMP. And most likely this is not going to work.
How long do you think it will be until we're at that point?
Well, if there are more efforts in the screening, and I hope that, how with your power, this will resonate even more in the world. So I guess that once these high throughput screening, they're called, once these high throughput screening are on the table and are known for, applied to ADMP, the sooner you can get a response on that. And so I think also there are the repurposed drugs as well, such as ketamine, for example, in low dose ketamine. I think you're all aware of this. But I mean, this is just taking one and apply that. But the high throughput screening is what will really make the difference. So you take all the compounds that you can possibly think of and say, let's see what happens in these cells. But these require massive efforts that we as a lab are not capable of. These will probably require pharmaceutical scale effort.
We've run the entire FDA approved drugs right now, and we have hits, but we don't have the cells yet at the level you have to start testing them.
Right. So, I mean, in the cells, it would be also interesting to test it, because if it's going directly to the patient, it would be also useful to test in the cells. I'm not aware of any lab that is doing that, but if there is some lab testing it,
we're
right now. Okay, yeah, no, that's
I'm going to be talking to you.
perfect. Yes, amazing. I absolutely.
Okay. No more questions. Thank you, Ludovico, for the nice talk and all the good questions. Okay, so then we can all go to lunch, the same place where we had the coffee break. Thank you.